Effects of APOE genotype on brain activity during movie watching, a pre-registration poster for the BNA Festival of Neuroscience 2021. APOE4 is the largest genetic risk factor for late-onset Alzheimer's disease. It has been associated with poorer episodic memory in older age as well as higher default mode network connectivity compared to E4 non-carriers during resting state fMRI. During movie watching, the default mode network has been shown to respond to narrative shifts, which can also be called event boundaries. These event boundaries have been suggested to be critical in helping to organize information in memory. Therefore, as E4 carriers show a difference in default mode network connectivity at rest, a network responsive to event boundaries, and potential differences in memory ability in older age, this study aims to investigate whether APOE4 carriers display unusual patterns of brain activity around event boundaries compared to non-carriers. This study will use data from the CAM-CAM dataset, which is a group of around 600 genotyped participants representing the adult lifespan. We will examine whether there are genotype differences in boundary-evoked responses to a short film. Each participant watched an 8-minute movie during an fMRI session. Data will be pre-processed using a pipeline developed by Geerligs, CAM-CAM and Campbell 2018. Using a predefined event segmentation of the film comprised of the 12 most salient event boundaries, we will test for differences in hippocampal responses to event boundaries and differences in default mode network functional connectivity around boundaries. We will select regions of interest from the default mode network and the hippocampus. Our analysis will be formed of three main components. Firstly, a general linear model will be fitted with event boundaries as a regressor to quantify boundary-evoked activity in each region of interest. A psychophysiological interactions analysis will be used to investigate the functional connectivity of the default mode network regions. This will be done by identifying if the correlation in activity between two regions differs as a result of the event boundaries. Finally, we will submit our results of these analyses to a mixed effects model with planned contrasts to compare differences between APOE genotype and age. Thank you to our collaborators of this proposed study and to the Alzheimer's Society and the European Research Council for funding this work. To see a more detailed outline of this proposed study, check out the poster PDF in the documents below. It would be great to get your feedback or any suggestions on the study, so do drop by the poster session to have a chat.